A phylogenetic review of the African Leaf Chameleons, focusing on genus Rhampholion in the family Chameleonidae. Chameleons are a group of organisms belonging to the family Chameleonidae of the class Reptilia in the phylum Chordata and the kingdom of Animalia. Chameleons are best known for their ability to change colors, their long, rapid-fire tongue, which is often up to 3 times the length of their body, as well as their independently mobile eyes, which can see in 2 different directions at the same time, or when focusing on a single object, allow them to see in stereoscopic vision. Some can even play the guitar. The subgroup of chameleons that we are interested in are known as African Leaf Chameleons or Pygmy Leaf Chameleons. They are typically very small, approximately 3 inches in length, and they lack the vibrant coloration of the other chameleon species. However, they do still have the ability to change color. It's just limited to a spectrum from light brown to black. Due to their ease of capture, the demand in the pet trade, combined with an extremely high mortality rate and often improper housing, has led to a large number of African leaf chameleons dying off. As a result of this, Tanzania, one of the primary countries of export, has closed their borders several years ago and has, as of today, remained closed for export. Although the family Chameleonidae is a monophyletic family, it is believed that the genus Bruchisia was the first to diverge from the other chameleon species. Followed by them, it was Rhampholion, the second group to diverge. A number of morphological and genetic studies have suggested that the Rhampholion genus should further be broken up into 2 subclades, type 1 and type 2. It is difficult to pinpoint the geographic origins of speciation amongst the chameleons in this study. This is predominantly due to the volatility of the geography of the region during the time period of divergence. Notably, the formation of the Eastern or Gregorian Rift and the Western or Albertine Rifts are believed to have influenced speciation dramatically. The specific statistical methods and software used are beyond the scope of our class. However, of note is that all Rhampholion species were used in this study, as well as including 1 sample of each chameleon genus as outlier groups. The data analyzed supports the previous notion of having 2 distinct phyletic groups within the Rhampholion genus. That is, type 1, as previously denoted, as well as a type 2 with 3 distinct subgroups within it. Furthermore, the molecular clock data and the Bayesian inferences show that Bruchisia likely diverged approximately 46.5 million years ago, which agrees with other data placing Bruchisia as a sister taxa amongst the chameleons. As a result of this data, it is suggested that a new genus be created, genus Ripelion, in honor of Dr. Ripel. All type 1 species are to be removed from the Rhampholion genus and moved here. Secondly, genus Rhampholion will consist of all previously existing type 2 species, having removed type 1 to the new genus. Furthermore, we suggest that we break this genus down into 3 subgenera, 2 as previously identified, as well as 1 new one, Rhinodigita. The full proposed tree with branch points labeled in millions of years ago is shown here. As can be seen, Bruchisia was the first to diverge off 46.55 million years ago. Next, it is suggested that type 1 Ripelion species are their own branch, denoted by the lowercase *b at approximately 11.10 million years ago. Type 2 is believed to be its own branch, further separated into 3 distinct groupings, grouping type lowercase I, lowercase ii, and lowercase iii, alternatively known as Rhampholion, Bicuspis, and the new group Rhinodigita. Taking this molecular phylogeny data that we now have and combining it with the geological data that we know, we can attempt to determine some hypotheses for the speciation of both type 1 and type 2 genera. Knowing that modern-day Rhypelion species are limited to the eastern rim of the African continent, combined with molecular clock data that puts its divergence at approximately 10 million years ago, relatively recently in the divergence timeline, does not leave much room for an explanation of the speciation between different Rhypelion organisms. Even if geographic flooding would have resulted in the loss of some lowland habitats, this would have left a single small population segregated, resulting in one lineage surviving once that habitat grew back out. It does not explain the existence of 3 distinct lineages. As such, their origins are not very well understood. This geological separation hypothesis is further supported by the molecular clock analysis, as the times of divergence correspond to the times of geological activity. Finally, molecular data further supports this hypothesis, as the subgroupings, as previously reported, groupings 1 through 3, also correspond to the geographic isolation and barriers created between them. Proposed genus Rhampholion, that is type 2, are believed to have diverged about 26 to 28 million years ago. This is almost 2.5 to 3 times longer than the believed time frame of type 1 divergence, which was 10 million years ago. During this extra time, Rhampholion species have had the chance to go through multiple diversification events, most notably occurring at 18 to 19, 11 to 13, and 6 to 7 million years ago. These were periods when the formation of the eastern and western rifts were created, as well as contraction and expansion of the Pan-African continent.